Okay, let's go. Question. Oh. Let's go ahead. This is the lab assignments that I've worked up. I just want to make sure that it's correct, at least in terms of who's in the 1 to 3 and who's in the 3 to 5 shift or section. So this is posted on Moodle, and then I more or less randomly divided the groups into sort of groups A and B in each of the each of the sections, each of the 1 to 3 or 3 to 5 sections. So for example, at 1 o'clock, group A will work with the cadaver, and group B will work with the pig hearts. And then at 2 o'clock, we'll switch. And then at 3 o'clock, group A will work with the cadaver, and group B will work with the pig hearts, and then at 4 o'clock we'll switch. Okay? I'll have a copy of this available in the lab if you forget whether you're in A or B. And you can, like I said, you can assign if your team wants to assign a name other than A, that's fine with me. You'll notice that one of the handouts is a list of key concepts that this is actually what we have been doing. So this is what we covered in the first couple of classes. And I will have a key concepts list for the cardiovascular section coming up, and I'll have that available to you on Tuesday. Okay, so I intend to have these key concepts lists at the beginning of each section, so you can sort of see where we are in terms of the key concepts, and then you will refer to it when you're preparing for the exam at the end of that section. Remember that there's a problem set due on Sunday, and I'm happy to answer questions about the problem set. You can bring them to lab or to my office hours tomorrow, or email me a question. I'm happy to try to make sure that those questions are clear. Okay. So today we're kind of wrapping up this whirlwind review of self-phys material, and going to start to transition into new material for the cardiovascular section. That's where we're going today. So we will start with muscle contraction, which is kind of where we ended on Tuesday. Then we'll get into specifics related to cardiac muscle and smooth muscle. Both of them are important for the cardiovascular system. So take what you know about skeletal muscle and apply it to these two other types of muscle. If you have a question about the problem set, maybe after the break, we can bring it to class. But like I said, in lab or office hours, they're also available. So here's a slide that we finished up with on Tuesday. It was getting so hot in here that some of you may have been not thinking. I know I wasn't thinking clearly, so let's just go through this again. In the relaxed state, when there is low calcium, intracellular calcium concentration, troponin and tropomyosin are in a configuration that occludes or blocks the availability of binding sites for myosin on actin. So myosin can't bind to actin, and so it doesn't. So the myosin heads and the actin filaments are separated. And the muscle is relaxed and can be stretched because the fibers are not connected to each other. When calcium rises, and we'll talk about the triggers for calcium rise in just a few minutes, but the calcium rise is the trigger for contraction. Intracellular or cytoplasmic calcium rises. Calcium binds to troponin. Conformational change. Troponin and tropomyosin get out of the way. Myosin now can and does interact with actin. That interaction causes myosin to let go of its bound ATP and phosphate. That ATP and phosphate, ADP and phosphate. I said it wrong. Let's go of its bound ADP and phosphate. That bound ADP and phosphate was there from the previous cycle from ATP hydrolysis. So the letting go of ADP and phosphate causes a conformational change of myosin from an extended to a bent configuration, and that, because it is bound to actin, it pulls on the actin. And the actin is attached to the Z discs, and so that pulls the Z discs together. That's the power stroke. Myosin has an intrinsic ATPase activity. Myosin ATPase. It hydrolyzes, I'm sorry, ATP has to bind first. ATP binds, and that ATP binding causes myosin to let go of actin. So ATP binding also causes a conformational change that reduces its affinity, myosin's affinity for actin, and so it lets go. Myosin also has intrinsic hydrolysis activity. It's an ATPase. When myosin hydrolyzes the bound ATP to ADP and phosphate, there we are, that ADP and phosphate came from that ATP hydrolysis. Now it goes back to the original configuration where, as long as calcium is high, it could bind to a new site on actin and do the cycle over and over again, bringing Z discs together. The trigger is calcium. Calcium grades the strength of contraction. So the more, and how is that? Calcium changes are almost never all or none. Calcium changes are graded. By graded, I mean, small, medium, large. It's a continuum of calcium concentration. The more calcium there is, the more sites on troponin are bound. The more sites are made available, the more sites on actin are made available for myosin, the more myosin can bind to actin, and the more and harder and faster the contraction can be. So contraction is graded by the amount of calcium. The strength of contraction is graded by the amount of cytoplasmic calcium. The whether or not the relaxation versus contraction is there a change in calcium, and then the level of contraction, the strength of the contraction is graded by the amount of calcium. Is that clear? Okay, so now let's apply, this is skeletal muscle. Let's apply what you know about skeletal muscle to cardiac muscle, which is very similar. Okay, so you know that skeletal muscle is considered striated, that is to say it has stripes. So if we go back to the cartoon, skeletal muscle has stripes under the microscope. It looks striped. So it's called striated muscle. Cardiac muscle is also striated because it has stripes. So like skeletal muscle, cardiac muscle has sarcomeres. That's what those stripes are, sarcomeres. I think I told you in Cell Phys that skeletal muscle forms from the fusion of the stem cells. So a skeletal muscle fiber, so for example, there's a skeletal muscle fiber in my biceps that extends from my elbow all the way to my shoulder. There's one fiber that is, inches long. And that is a big cell because it was formed from the fusion of precursor cells. So that's why skeletal muscle is multinucleated. Each muscle fiber cell has multiple nuclei, one from each of the precursor stem cells that fused to form the big fiber. Cardiac muscle isn't like that, is not like that. Cardiac muscle cells are not fused. Each cell has its own nucleus, just like a normal cell. But cardiac muscle cells, so this is supposed to be a cartoon of a junction between two cardiac muscle cells, this interdigitating membrane making a connection between two muscle cells. So the cardiac muscle cells are individual cells, each with their own nucleus, each with their own plasma membrane. But the plasma membranes of adjoining cardiac cells are connected. They're connected physically with this structure like that, at a structure called the intercalated disc. So they're connected physically, so if one muscle cell contracts, it pulls its neighbor with it. And they are connected electronically through gap junctions at a structure. So the intercalated discs have a substructure called the desmosome. I'm not gonna make you memorize these names, but I do want you to know that cardiac muscle cells are connected physically and electrically. The gap junctions are pores that connect one cell to the next cell. So a gap junction goes through two membranes, the membrane of one cell and the membrane of the connected cell, so that the cytoplasms of the two cells are connected electrically through this ion channel. The key thing to know is that if one cell fires an action potential, one cell depolarizes and fires an action potential, its connected neighbors will also depolarize and fire an action potential because they are connected through these gap junction ion channels. That's the key thing. We need to know that in order to understand the heartbeat. To repeat, cardiac muscle cells are electrically and physically connected to their neighbors. Now let's go back to calcium. You'll recall from cell physiology that the surface membrane of muscle, especially striated muscle, has structures called T-tubules. The surface membrane has these penetrations deep into the cytoplasm called T-tubules. So, and these T-tubules, but these T-tubular membranes are, part of or extensions of the surface membrane. So any electrical event, like an action potential propagating on the surface of a muscle cell, propagates into the T-tubule. So when a skeletal muscle fires an action potential, let's get relevant. When a cardiac muscle fires an action potential, that action potential propagates into the T-tubule. In the T-tubule, in the membrane of the T-tubule, there are channels. There's a voltage-gated calcium channel in the T-tubular membrane. So when an action potential propagates from the surface of the muscle into the T-tubule, it opens a voltage-gated calcium channel. And which way does calcium flow? How many say calcium flows from the extracellular space to the cytoplasm when a voltage-gated calcium channel opens in the T-tubule? How many say that calcium flows from the cytoplasm? So here's the channel, this little red dot. Calcium can either flow from the space in the T-tubule, which is connected to the extracellular space, into the cytosol, or from the cytosol into the extracellular space. You need to know the gradient for calcium. Where is calcium concentration high, and where is calcium concentration low? High outside, low inside. So which way does calcium flow? Flows into the cytosol. Calcium flows into the cytosol and triggers a nearby channel called the calcium release channel to open. So calcium flowing through the channel in the T-tubular membrane triggers calcium channels in the SR membrane, and calcium flows out of the SR. So remember that in the SR, calcium concentration is high. Why? Circa. You got circa pumps calcium into the SR. So the calcium concentration is high here. The calcium concentration is high here because of the plasma membrane calcium ATPase. When the voltage-gated channel opens, calcium flows in. There's calcium influx. And that calcium influx triggers a calcium-induced calcium release channel in the SR to open, and more calcium flows, this time, from the SR to the cytoplasm. Both of these are essential for a cardiac muscle contraction. Jasmine?
So the calcium release channel is in the SR, or?
It is in the membrane of the SR. Okay. It's in the SR membrane. Okay. Channels are almost always in the membrane. So, just to review the story here, an action potential propagates into the T-tubule. That action potential opens a voltage-gated calcium channel. It's called the, so names are, we can either give it a pharmacological name. So if I wear my pharmacologist hat, I call it a dihydropyridine receptor, because it happens to be the protein that a class of drugs that is used clinically for heart disease called dihydropyridines binds to. So when I put on my medical or pharmacological hat, I call it a DHP, or a dihydropyridine receptor, because that's the protein that is affected by that drug. If I put on my physiologist hat and I try to ignore the pharmacology, I call it a voltage-gated calcium channel. It's the same thing. It's just what do I want to call it? I'll accept both names. You can call it a DHP receptor, or you can call it the voltage-gated calcium channel. It's the same protein. So the action potential, a large depolarization, opens a voltage-gated calcium channel, also known as a DHP receptor. Calcium flows in, down its concentration gradient. That influx of calcium is received by a calcium release channel on the surface membrane of the SR, which is also called, if I'm a pharmacologist, I call it the ryanodine receptor. Ryanodine is a class of toxin, really. It's not really a drug. But the calcium triggers the calcium release channel to open, and more calcium flows, this time from the lumen of the SR into the cytoplasm. Both of those are important for the rise in intracellular calcium. And now we're off and running with the contractile mechanism that I told you about in the previous slide, which I don't have to review because you know it. More calcium, more contraction. At the end of a contraction, we need to relax. Your heart needs to relax between heartbeats. So we need to take up the calcium, and that's what the plasma membrane, that's what PMCA and CERTA do. To get rid, to lower intracellular calcium between heartbeats so that the heart can relax and refill. You gotta have it, it's gotta be relaxed between beats. You can't just contract once or Me, did you have a question?
Yes, I missed the part that you said. What triggers the ryanodine receptor?
What triggers the ryanodine receptor? Calcium. Calcium from the dihydropyridine receptor. I'm using the pharmacological names, but I could also use those physiological names. Jasmine.
So PMCA and CERTA pick up calcium from the cytosol, and they lower the levels so that the cycle can start over again?
Exactly.
Okay.
And PMCA pumps the calcium outside the cell, and CERTA pumps the calcium into the SR. But both of those lower cytosolic calcium so that the heart can relax between heartbeats, so that the next heartbeat actually can work. Yes.
In this case, the ryanodine receptor trigger the release of kind of trigger or
Yeah.
open the calcium release channel, then in G2 coupled pathway, is IP3 does the same role?
So this calcium release channel is similar to the IP3 gated calcium release channel from the SR, but is not exactly the same. So in cardiac muscle, the more important mechanism for calcium release from the SR is this calcium-induced calcium release. When we get to smooth muscle in just a few minutes, IP3-induced calcium release is going to be important. This is similar to the IP3-induced calcium release of the GQ story, but it is not the same as the IP3-induced calcium release of the GQ story. Good question. Is this clear? What is similar to skeletal muscle? The mechanism of contraction. The fact that there are T2s. The fact that calcium grades the what's different? There's a couple of similarities, there's a couple of differences, but I don't want to confuse the issue. A couple of differences. Yes, John.
What's the channel called that takes the calcium back into the SR?
We don't call it a channel. We call it a pump because it is pumping calcium up its concentration gradient. And channels don't do that. Channels only allow calcium to flow down their concentration gradients. The pump that pumps calcium into the SR is called SERCA. Sarcoplasmic endoplasmic reticulum calcium ATPase. It's very important for us to have these conversations so that I can make sure that I'm speaking your language and that you're speaking mine. Other questions? Okay. That's skeletal muscle, cardiac muscle. Well, here's a cartoon that tries to show, I probably should have shown this on Tuesday, but here is the thick filament. Here are the purple are the thin filaments. The red is the thick filament. The zigzag here is the Z disc. The idea here is that when calcium increases and these motors, the myosin motors, are pulling on the thin filaments on each side, the sarcomeres from here to here are getting shorter. And that's contraction. That's the same in skeletal muscle as it is in cardiac muscle. The motor activity of myosin binding, letting go of its ADP and phosphate, power stroke, pulling, that was a pull. I just pulled the two Z discs closer to me. ATP binds, I let go. ATP hydrolyzes, I go to the extended configuration. Calcium is still high, so I can bind again to a new spot. Because it didn't stretch. So a new spot, and I do it over and over again, and I pull the two Z discs of the sarcomere closer together. The muscle shortens, the muscle contracts. And if you're a heart muscle cell, you generate pressure to force blood to flow. We'll get to that in the next week or two. Okay, let's do a quick one. looking for the best answer or answers for the specifics of what is required for myosin to bind to actin. Not thinking about what happened before, and I'm not thinking about what happens after. I'm thinking about what allows myosin to bind to actin. What do you need in order for myosin to have the configuration that gives it high affinity for actin and causes it to bind? Okay, so this is pretty hard, just based on the number of correct answers that I'm seeing. It's A and C. Both A and C are good. So calcium is required. Why? To move the troponin and the tropomyosin out of the way, because otherwise myosin can't bind to actin. ADP is required in order for the ADP to be there, because if it isn't there, it's in a configuration that doesn't have a high affinity for actin and myosin. I mean, it doesn't give myosin a high affinity for actin. So ATP is not a correct answer. Why? Because if ATP is necessary for it to unbind, not to bind to actin. That's the idea. Now,
So when you say cytoplasmic ADP, is that the ADP bound to the?
yeah, so the idea
and
is.
not free in the.
So I could see, you could say, oh, it's not cytoplasmic ADP that's necessary. You need to have ADP bound to myosin. And I could see why that would be
Okay.
confusing. But remember that binding is always in equilibrium. Always. There's always some form of equilibrium. And if we had no cytoplasmic ADP, none, if we had no cytoplasmic ADP, then all of the ADP that was bound to myosin would come off eventually, because it's in equilibrium. So it's true. When I think of what's necessary, I'm always remembering a binding equilibrium. When things are bound, it's never permanent. It's never. It's always in an equilibrium condition. The number of times where we're not. I mean, we're not at equilibrium, but there is binding and unbinding going on. So I could see why that could be confusing. Okay, so let's do the next one. Which of the following is required for the power stroke? Best answer is E. The unbinding of ADP and phosphate is what triggers the power stroke. So you'll work through this. I think, I can't remember if there's a question that asks you to use this in your problem set, but if not, you should have the story of the molecular mechanisms for muscle contraction sort of in your head. Questions about that one? I think most of you got that one. Okay, so let's turn our attention to smooth muscle. Why do we have to talk about smooth muscle? Well, smooth muscle lines almost all of the tubes in your body. And in particular, for this section of the course, we need to think about the control of the diameter of arteries and veins. Right? Arteries and veins are part of the cardiovascular system. You can't just focus on the heart. So smooth muscle, which lines the arteries and veins, can contract or can relax, and that controls the diameter of the tube, which is important for blood flow. We'll get to that in a few minutes. So smooth muscle is important for the cardiovascular system. It's going to be important for controlling the diameter of the airway when we get to the respiratory system. It's going to be important for controlling the contraction of the urinary bladder when we get to the urinary system. It is important for the contraction of the uterus in the reproductive system, which we're not going to talk about in this course. It's going to be important for the constant contraction of muscle surrounding the digestive tract, from the esophagus down to the colon. Smooth muscle is going to be controlling the movement of material in your digestive tract. So we're going to be seeing it over and over and over again, so let's just get it out there. Smooth muscle is not striated, So it doesn't have Z discs and it doesn't have T tubules. So it's unlike cardiac muscle. But like cardiac muscle, smooth muscle cells are connected by gap junctions. So when one smooth muscle cell contracts, its neighbors get, or at least when one smooth muscle cell depolarizes and gets calcium and contracts, its neighbor is going to do the same thing. So it's coordinated kind of the same way that cardiac muscle is. Like cardiac muscles, your smooth muscle is not typically under your voluntary control. I mean, you can control your skeletal muscle. You can't really control your heart muscle. I mean, I can control your heart muscle. I can say, I've got a quiz for you, and this is going to count for 20% of your course fee, and I can get your heart muscle to contract a lot more forcefully. But you see what I'm saying? The sympathetic nervous system controls the contraction of the heart muscle, but it's not really under your conscious control. And the same is true for smooth muscle. You can't control the diameter of your arteries or veins. You can't control the contraction of smooth muscle in your digestive tract. Your nervous system does control those things, but it's not part of your voluntary mechanism. So smooth muscle, so this looks like a cartoon, but this is actually a micrograph of smooth muscle. Each of these blue things is a nucleus. The pink thing is a smooth muscle fiber. It looks like they're all separated, but there are gap junctions between different cells. The main point is that smooth muscle comes in circular and longitudinal structures around the tubes that it controls. So if this is the tube, we have some smooth muscle that's oriented circularly around it that controls its diameter, and other smooth muscle that is organized longitudinally that controls its length. And both of those are going to be important. So smooth muscle is organized longitudinally and circularly around the tube that it controls. I said they don't have T tubules. They don't have Z discs, but they do have connections. So there is myosin and there's actin. It does the same thing as skeletal and cardiac muscle in terms of the contractile mechanism. So the thin filaments are not attached to the Z discs, so there are no Z discs, but the thin filaments are attached to these things called, I forget what they're called, dense bodies. You don't have to know that name. But the thin filaments are connected to the membrane, and then there's myosin in between there. And so when the cell is triggered to contract, what do you suppose triggers the contraction of a smooth muscle? Take your best guess. It's going to be the correct one. Calcium. A rise in intracellular calcium. That triggers myosin to pull on these actin filaments, and so the filaments get shorter, and the muscle falls up, gets shorter. It's not as highly organized as it is in skeletal or cardiac muscle, but it's the same idea. So remember, if a muscle that is part of a circular organization, if those muscles contract, the muscles get shorter, the diameter of the tube gets smaller. That's vasoconstriction. If the smooth muscle relaxes, then the pressure inside that tube can stretch the muscle, and the tube gets dilated, and that's vasodilation. Contraction and relaxation of the circular smooth muscle controls the diameter of tubes, and especially the arteries and veins that we were talking about this. Okay? Questions about smooth muscle? So like I said, the thing that causes smooth muscle to contract is cytosolic calcium concentration. Again, it's graded. Higher concentrations of cytosolic calcium, more contraction. Less concentration of cytosolic calcium, less contraction. Very, very low concentration of cytosolic calcium, dilation or relaxation. Based on this slide, is troponin the thing that calcium binds to the way it is in skeletal and cardiac muscle? No, it doesn't look like it. What is the calcium sensor in smooth muscle? Calmodulin. Remember when I taught the GQ story that I told you about? And I said calcium, that in the book, the GQ story, the IP3, the GQ, phospholipase C, part of your homework assignment, the calcium, the book focuses on calcium activating PKC, but I added to that slide and said, don't only think about PKC, also think about calmodulin. Here we go. Here's calmodulin. Calcium binds to calmodulin, activates calmodulin. Calmodulin activates a kinase called myosin light chain kinase. That's one of the things it does. There are other kinases. Some of you may have heard of calmodulin-dependent protein kinase 2, CAMK2. Anybody work on a summer research project related to CAMK2? It's pretty common. Okay. Never mind. In this case, the thing that calmodulin activates is myosin light chain kinase. Myosin light chain kinase phosphorylates myosin. So in this case, it's a phosphorylation of myosin that causes it to have high affinity for actin. It's not, there's no troponin. It's not the troponin, tropomyosin getting out of the way that's the trigger. The trigger is whether the myosin is itself phosphorylated or not. Bottom line, cytosolic calcium activates calmodulin. Calmodulin activates the kinase. Kinase activates myosin, and you get the cross-bridge cycle that you already know about, and the smooth muscle balls up and gets shorter. So some of you could be wondering, okay, fine, great, whatever. How did we get this increase in cytosolic calcium? I'd like you to take what you know about G protein coupled receptors and cardiac muscle and give me two ways that smooth muscle could increase cytosolic calcium when it is triggered to contract. Remember, there's a trigger. What are the two pathways? There are two pathways. Yeah, two pathways that could trigger an increase in cytosolic calcium. One of them I'm going to ask you to think about from Tuesday, and one of them I'm going to ask you to think about from just a couple minutes ago. Anybody want to venture? I'm asking you to read my mind a little bit. So anybody want to try to read my mind? Me.
Again, slide one of the ways we have the
That's okay. Then there's three. That is a good way. So if a neighboring cell had an increase in calcium, then this cell that is completely correct. That is completely a correct answer. I wasn't thinking of that, but that is correct. So if a neighboring cells had an increase in calcium, then that calcium would flow from one cell to the next cell through the gap junctions, and that would trigger this cell to contract. So that is one way. Completely correct. Now let's see. Try two that I was thinking of. Yes, Zoe.
Are you thinking about the
Exactly. I'm thinking about the activation of the GQ pathway. So what's not shown on this slide, but which you should probably decorate this slide to show that the surface membranes have GQ coupled receptors. Right? Because if a drug or a hormone or a neurotransmitter activated a GQ coupled receptor here, let's tell that story. Activation of receptor conformational change, alpha Q conformational change, GDP dissociation, GTP association, alpha separating from beta gamma, alpha, the activated alpha with GTP bound activating phospholipase C, hydrolyzing PIP2 to IP3 and diacylglycerol, IP3 going to the SR and releasing calcium from the SR, and now we have our calcium. So we've just told one of the two stories that I was thinking about. So now we have two ways that calcium could increase. Activation of a GQ coupled receptor, or calcium coming in through a gap junction. One more.
Last year, but can we inhibit
We could, but that's not right. It's a good thought, but that's not one of the things that triggers contraction. It just doesn't happen. Yeah. Sure. Exactly. Voltage-gated calcium channels. So that is the answer. On the surface membrane of smooth muscle cells, there are voltage-gated calcium channels, very similar to the voltage-gated calcium channels that are in the T-tubule of cardiac muscle. So anything that triggers an action potential on the surface membrane of smooth muscle would trigger a voltage-gated calcium channel, opens, calcium flows in, and there we have an increase in cytosolic calcium. Different smooth muscles have different levels of voltage-gated calcium channel dependent action and GQ coupled receptor action. So airway smooth muscle is a little different from artery smooth muscle, but and I'll get into those specifics as needed. But I want you to think about the three, thank you, the three ways that a smooth muscle cell could get triggered to contract. And the fact that it's graded, right? Why would it be graded? More calcium, more active calmodulin, more active kinase, more cross-bridge cycling. Yes?
Then what are the channels that are like located in gap junction? Are they with calcium?
This is a good question. What are gap junction channels calcium channels? Not really, because they're not specific. They allow almost anything below a certain size to flow. So they allow sodium to flow, they allow potassium to flow, they allow calcium to flow, they allow chloride to flow, they allow phosphate to flow, they allow ATP actually to flow from one cell to another. They're non-specific holes that connect the connect the cells. Do they allow proteins to flow? No, their proteins are too big. But they allow all the small molecules, including ATP or cyclic AMP, and also including calcium and potassium and magnesium, to flow from one cell to another. So are they calcium channels? Yes and no. They allow calcium to flow, but they're not specific for calcium. Any other questions? Let's take a break, and then we'll transition to this overview of the cardiovascular system. All right, let's do the heart. Let's start talking about the cardiovascular system, and we're going to start with the heart. The heart is really two pumps. There's the left side and the right side. Two pumps in series. So we talk about the left heart and the right heart. The left heart is comprised of the left atrium, so one atrium, two atria. It's just the words. The left atrium and the left ventricle form the left heart. We only have one heart, of course, but the left side of the heart is really its own unit, and the left atrium and the left ventricle form the left side of the heart. The right atrium and the right ventricle form the right side of the heart. The atria are superior and the ventricles are inferior in the heart. You're going to be seeing this in the lab today. I'm just orienting you. The atria, just like the atrium of a building, is the receiving area. The atria on the right atrium receives blood from the body. The left atrium receives blood from the lungs. The atria are connected to the ventricles through valves that I'll give you names of in a minute, not today. That's part of the anatomy that you're going to learn. Well, I am going to use some of the names. They're not yet. Blood flows from the right atrium into the right ventricle, and blood flows from the left atrium into the left ventricle. When the heart contracts, muscle contraction, blood is pumped from the right ventricle into the pulmonary circulation, to the lungs. So the right ventricle pumps blood to the lungs. The left ventricle, when it contracts, it pumps blood to the body. So we have to think about two different circulations. The circulations to the lung, called the pulmonary circulation, and the circulation to the body, which is called the systemic circulation. Pulmonary circulation is blood flow to the lungs, and systemic circulation is blood flow to the body. Now you know that you have arteries and veins, and so there's arterial and venous circulation. Blood in the arteries is arterial. Blood in the veins is venous. Just using venous with a O. So let's walk through the systemic circulation. The left side. Blood flows from the left ventricle. I should say, blood is pumped from the left ventricle into the aorta. Aorta is connected to major arteries, some of which you will learn the names of, but not all of them. So arteries to major systemic arteries, which have names. And those major arteries are connected to a network of smaller and smaller arteries, eventually getting into these little tiny things called arterioles. So the aorta is a big tube. Systemic arteries are pretty big tubes. And then they branch and they branch and they branch and they branch to these tiny little tubes, because now we have blood basically in contact with cells. So we talk about the major arteries, and then this is the micro circulation. These are microscopic in size. Arterioles, capillaries, and venules. So blood is flowing this way, through arterioles, into the capillaries. What happens in the capillaries? Let's just get ahead of ourselves for a moment. What happens in the capillaries? Somebody? I heard something. I heard right things. Now, gas exchange. So oxygen is being delivered from the blood to the tissues. Nutrient exchange. Glucose is being given from the blood to the tissues. Carbon dioxide, more gas exchange. Carbon dioxide is being absorbed from the tissues into the blood. So gas and nutrient exchange is the short answer to what happens in the capillaries. And that's why we have blood, is to provide gas, oxygen, let's go back to self-is. Why do you need oxygen? Why are we breathing oxygen? It's the terminal receiver for electrons in the electron transport chain. So then the microcirculation, that's it, trying to illustrate by this network here in your textbook, joins into small venules, which then collect, merge to form larger veins, some of which you have the name of, like the jugular vein or the cephalic vein. There's a vein here in your shoulder that we'll see in the lab. Some of these. Let's see. Can you think? Do any of you already know the name of one of the major arteries? What you do? What's the name of the major artery in the neck? Carotid artery. What's the name of the major artery in your arm that they measured blood pressure from? Anybody know that one? Brachial. Yeah. So those arteries have names that you will learn as part of the anatomy part of this course. And you know some veins: jugular vein, cephalic vein. We'll do more of that. So the big veins and the big arteries have names, and the little ones that are microscopic don't. Or if they do, I'm not expecting you to know them. Then the systemic veins join into the largest vein of the body, called the vena cava. And the vena cava returns now deoxygenated blood from the body, because the body absorbed all that oxygen and nutrients in the capillaries. The deoxygenated and the denutriated the nutrient poor blood and the deoxygenated blood return to the heart, to the right atrium, and then go to the right ventricle. So that's an overview of the systemic circulation. Let's do the same thing for the pulmonary circulation. Deoxygenated blood from the right ventricle is pumped into a big tube that has a name called the pulmonary trunk. And the pulmonary trunk divides into pulmonary arteries. There's names. And those pulmonary arteries go to the lungs, where the microcirculation exists. Arterioles, capillaries, and venules. And at capillaries, what happens? Gas exchange. But now we're picking up oxygen and getting rid of carbon dioxide. By we, I mean our blood cells, right? The red blood cells. Then the microcirculation in the lungs collects into venules, and this vein, it's called the pulmonary vein, brings the blood from the lungs to the left ventricle. Now there's a confusing thing. Wait a second. Isn't arterial blood oxygenated and venous blood deoxygenated? That's true in the systemic circulation, but it is wrong in the pulmonary circulation, because the correct definition of an artery is a tube that takes blood from the heart. So the aorta is an artery that takes oxygenated blood from the heart to the body. The pulmonary trunk is an artery that takes deoxygenated blood from the heart to the lungs. Okay? It's not really confusing if you think about the way the blood is flowing. It's only confusing if you use one of these color schemes. Okay? So the aorta is the biggest artery of the body. The pulmonary trunk is probably the second biggest artery of the body. The vena cava is the biggest vein of the body. And the pulmonary vein is probably the second biggest vein of the body. But you notice that the vena cava is carrying deoxygenated blood, and the pulmonary vein is carrying oxygenated blood. Is it clear? Arteries are taking blood away from the heart. Veins are taking blood back to the heart. Okay, so I'm just introducing some of the terms. So the output of the heart, the amount of blood being pumped out of the heart is called the cardiac output. And it's in units of liters per minute or milliliters per minute. You could convert that into liters per second or milliliters per second or liters per day or whatever. You could do the units conversion, but the typical conversion is liters or milliliters of blood per minute. And a typical value is something like 5 liters per minute. 5 liters of blood every minute is being pumped by your heart. There's a problem set question. I think the last problem set is going to ask you to do some unit conversion and some, sort of simple physical calculations based on cardiac output. Cardiac output is the flow of blood from the heart. And then that blood, some of it goes to different places, right? So some of it goes to the digestive tract. And some of it goes to the skeletal muscle. And some of it goes to the kidneys, right? So each of these areas is supposed to illustrate a different area that the blood is going to. It's divvied up. And some of it is going to the brain. We don't even see that. The brain is above the lungs, but so on this picture, a lot of it actually is going to the brain. So the cardiac output is divvied up based on the diameter of the tubes, really, that carry it to these various areas, whether it's kidneys or skeletal muscle or digestive tract or brain. So right now, hopefully, a lot of the blood that's coming out of your heart is going to your brain. Not as much as going to your skeletal muscle, because you're not exercising right now. But if you, after this class, you decide you're going to take a 1K run, before lunch, then more blood is going to go to your skeletal muscle because it's demanding more oxygen. So where the blood goes depends on the circumstances, and we'll get into how that's controlled in a couple of days. But here are typical values. So on average over time, approximately 13% of the cardiac output goes to the brain on any given time. And so when you're sleeping, it's a little bit less. When you're taking an exam, it's a little bit more. 4% of the blood from the heart is actually going to the the coronary circulation to actually feed the heart itself, right? All of the blood is going through the heart, but some of it is actually only 4% of it is actually feeding the heart itself. 20% on average goes to your muscle, 9% goes to your skin for thermal control, right? In order to release heat that your body has generated. 20% is going to your kidneys for processing of toxins, getting rid of waste products, and other things that the kidneys do that we'll talk about in this course. 24% is going to your digestive tract. 10% or so is going to everything else. Okay? Do I want you to memorize these numbers? Absolutely not. No. I'm just illustrating this. You don't even think about memorizing any of these numbers. Okay? You don't even have to memorize 5 liters per minute as the cardiac output. I'll always give that to you. So this is the division of blood in the systemic circulation. Brain, muscle, kidneys, digestive tract, a few others. What percentage, let's go back. What percentage of the cardiac output goes to the lungs? I'm not talking about feeding the lungs, I'm talking about going through the lungs. 100%. Right, that's a trick question that when I used to teach in a medical school, that was the favorite trick question of medical students. But you picked up on it remarkably fast. Very good. 100% of the cardiac output goes through the lungs. Because, right, all of the blood that came out of the aorta had to come from the lungs, right? They're in series. If we have a mismatch between the amount of blood going through the lungs and coming out of the heart, blood is building up somewhere, and that's usually a bad sign. Okay. So let's talk about pressures and volume. So we've talked about flow. Just be clear, we've talked about this is blood flow to the various organs of the body. And this is blood. Now let's talk about pressure. Pressure and flow are related. The left side of the heart, well, the left side of the systemic circulation is under high pressure because the left ventricle, and you're going to see this in lab today in both the pig and the human heart, the left ventricle is very muscular. And when it contracts, it generates a lot of pressure. And that lot of pressure is what's necessary to force that blood to flow to the brain and the rest of the body. You need high pressure on the left side of the systemic circulation in order for blood to flow. And so the left side of the systemic circulation is high pressure. And now, at any given time, about a quarter of the blood is in the high pressure side. So you'd like to think, the cartoon, oops, here's the cartoon. The cartoon makes it look like it's half and half. And that's not right. 25% are in the high pressure side, and 75% is on the low pressure side. So if the left ventricle generates the pressure, which causes blood to flow, remember, blood is flowing down its pressure gradient. So that means there's low pressure on this side and high pressure on this side. So the veins are under low pressure. This is why when you give blood, they try to puncture a vein, not an artery. Hopefully you've never seen what happens if you puncture an artery. The blood spurts because it's under high pressure. They don't want to do that. When they draw blood from your body, they draw it from the low pressure side so that there's not blood spurting. It's just flowing into the thing that they want to collect the blood in. So low pressure in the veins. And it's a lot lower in pressure, but it's higher in volume. The left side, or the left side of the systemic circulation, is high pressure, but relatively low total volume at any given time. And the right side, or the low pressure side of the systemic circulation is, and we're talking about systemic, we're not talking about pulmonary. Systemic circulation has about 3/4 of the systemic blood. The pulmonary circulation is kind of separate. So that's not part of this equation. Do I want you to know any of these numbers? No. I just want you to know that the high pressure side has less blood than the low pressure side. 1/3 as much.
Sorry.
Yeah.
So, are you saying the systemic is the left?
The systemic circulation is from the left heart, from the left side of the heart. So when I say left side, I'm talking about the left heart, which is generating high pressure. The venous circulation is connected to the right side of the heart. The right side of the heart is not pumping this way, it's pumping out this way, but we're ignoring, for this purposes, the pulmonary circulation. Okay? So let's just focus on systemic arterial versus venous arterial. Arterial, venous. So here's a couple of slides that show when I talk, remember I mentioned that arteries are connected to arterioles and there's intense branching. Well, here's a slide that shows the branching, the intense branching of the coronary circulation. So this is the heart. This is a major blood vessel that comes off the aorta called it's one of the coronary arteries. You've heard of that. Coronary artery disease is very important because if you restrict blood flow in the coronary artery, you restrict blood flow to the muscle of the heart, not the blood flowing through the heart, but the blood flowing to the muscle of the heart to feed it. And if the heart muscle starts to get starved of its nutrients, you've got a heart attack. Anyway, this is a coronary artery. Here is the left ascending, the left anterior descending artery. Another coronary artery that has a name. And you can see all of these little guys branching off of it, feeding the heart muscle itself. So the point is that this major artery dividing into these 2 or 3 different other secondary major arteries, branching into millions of microscopic arterioles and capillaries. Let's talk about the size of these tubes. So, a tube has a cross-sectional area, right? Pi r squared. The area of a circle. Right? Some of you might hopefully remember this from grade school. So the diameter, or the cross-sectional area of the aorta, which we'll probably see in lab today, is about 4 square centimeters. When it divides, so the diameter of all of the major vessels that it is attached to is slightly more than 4 square centimeters. Okay, so maybe it's 20 or something. The point is, when we get into the microcirculation with those millions and millions and millions of tiny vessels all connected to each other, each one of them is microscopic in diameter, but we've got millions of them. And so the total cross-sectional area of that bed of millions and millions of capillaries is actually huge. So even though each one is microscopic in diameter, the total, because there's so many of them, billions probably, I don't even know, because there's so many of them, the total cross-sectional area of a capillary bed is 3,000 square centimeters compared to 4 square centimeters for the aorta itself. You need to know you need to know that number or something about it for the problem set. You'll use this concept in the problem set. The area of the pulmonary artery, like the cross-sectional area of the aorta, is low. I mean, it's a big tube, but there's only one of it. And then it branches and branches and branches into billions of pulmonary capillaries, which have a huge cross-sectional area. The main point is that even though the capillaries are microscopic in diameter, their total summed cross-sectional area is very large. And that's what gives them this large surface area to interact with every single one of your cells. You need that large number of them and their small size in order to do the gas exchange and nutrient exchange that your body needs. Questions about this? It's not exactly intuitive. You think of the aorta as being a big tube. You think of a microcirculation as being a tiny tube. What doesn't come naturally is to think of what happens when you take a billion of those tiny tubes and look at them together, and it makes a huge total cross-sectional area. It's hard to think about billions, no matter what they are. Billion seconds, billion billion dollars. Questions? I think I'm done. Yeah, we'll talk about blood flow in the heart next week. What your goal for this afternoon is to explore the structures of the heart. And then we're going to take the anatomy that you're going to learn in lab today and the physiology that I'll teach you next week to put together the heart piece. Today's goal is anatomy. Any questions? Okay, so go enjoy some lunch, take a short break. Some of you I'll see at 1 and the rest of you I'll see at 3.